Hello everyone, this is your educator Priyanka Thakur. I'm teaching you life sciences and welcome you all on our YouTube channel Gurmantra Shikshaka Granth. So today this is your phytohormones part 4 and I'm going to tell you about the ethylene phytohormones. I'll be telling you about its biosynthesis, its different physiological effects and also its signaling pathway. Okay, I haven't covered the signaling pathways of auxin, gibberellin, cytokinin or abscisic acid, but today I'm going to cover the signaling pathway of ethylene. So let's start with our today's phytohormone, which is ethylene. Ethylene is a gaseous hormone and it is only member of its class, just like abscisic acid. Abscisic acid is the only member of its class. Okay, now just like that, ethylene is also only member of its class. It has simplest structure among all the known plant hormones. And mainly this hormone is associated with ripening of fruits and triple response. In later section I will tell you what is triple response. I haven't included this thing in my slide, but in 1910 cousins he studied that he saw that bananas were ripened when they were placed along with oranges. According to him, there must be some volatile substance which diffuses out of oranges, and due to that volatile substance bananas were ripened. Why? Because he bought bananas and then he placed some of the bananas besides oranges, but others were far away from the oranges. Okay, so those which were not beside the oranges were not ripened. Okay, but while the bananas which were placed along with the oranges, those were ripened. That's why I concluded that there must be some volatile substance which is responsible for the ripening of these bananas. In 1934, Gain was able to report that a plant synthesized ethylene and he reported it with the help of gas chromatography. After 1 year of this report, Crocker in 1935 proposed that ethylene was the plant hormone responsible for fruit ripening as well as senescence of vegetative tissue. Ethylene is produced almost all the parts of higher plants, including leaves, stem, roots, flowers, fruits, tubers, seeds, etc. Okay, but its production is regulated by a variety of developmental and environmental factors. Ethylene can stimulate transverse or isodiametric growth. See, isodiametric growth means the growth pattern will have same diameter in all of its sides. Okay, so ethylene can stimulate the isodiametric growth or transverse growth, but it retards the longitudinal growth. Fine. Okay, now see, let's talk about biosynthesis of ethylene. Methionine is the precursor molecule responsible for the synthesis of ethylene. So you must have to remember the precursor molecule of abscisic acid, cytokinin, gibberellins and auxins. They can ask you a question in match the following, and you have to match the correct precursor of correct phytohormones. So methionine is the phytohormone for ethylene. Then what is the phytohormone? Methionine is the precursor for ethylene. Then what is the precursor of auxin and gibberellin and cytokinin and abscisic acid? Okay, see, methionine is converted into 5-adenosyl methionine, or also known as S-adomate or SAM. Methionine is converted into SAM in the presence of enzyme adomate synthetase. Okay, Adomate synthetase is responsible for this conversion, and then this adomate or SAM is converted into 1-amino cyclopropane 1-carboxylic acid in the presence of ACC synthase. This is ACC, amino cyclopropane 1-carboxylic acid, and ACC synthase is responsible for this conversion. And in the last step, this ACC, with the help of ACC oxidase, is converted into ethylene. Fine. So we reached in our last step. What is happening here? Here, ACC is getting converted into ethylene in the presence of ACC oxidase. Let's revise these steps again. Methionine, okay, close your eyes. Methionine will be converted into 5-adenosyl methionine, also known as adomate or SAM. It will be converted with the help of adomate synthetase. Okay, there will be the release of energy will be used, not release, energy will be used. Okay, and then this SAM is converted into 1-amino cyclopropane 1-carboxylic acid with the help of ACC synthase. And in the final step, oxygen will be utilized with the release of carbon dioxide and hydrogen cyanide, and the enzyme responsible for this conversion of ACC into ethylene is ACC oxidase. Okay, the biosynthesis of ethylene is also known as Yang cycle. This is because this S-adomate have the capacity, okay, this adomate have the capacity to get converted back into the methionine. Okay, get it? This is Yang cycle. Okay, S-adenosyl methionine or S-adomate or SAM will be converted back into the methionine with the help of some intermediate enzyme, but the cycle is called Yang cycle. This step in which S-adomate is converted into ACC or 1-amino cyclopropane 1-carboxylic acid. This is a step mediated by ACC synthase. It can be inhibited by amino ethoxy vinyl glycine and amino oxyacetic acid. So these are the inhibitors of biosynthesis of ethylene. Which one? Amino ethoxy vinyl glycine and amino oxyacetic acid. These both are the inhibitors of biosynthesis of ethylene, and they inhibit the conversion of S-adomate into ACC. Okay, remember, auxin promotes the ethylene synthesis. And how the ethylene is transported? Ethylene can be transported by diffusion because it is a gaseous hormone. It is one of its own kind. Okay, it is one of its kind. Okay, so it can move by diffusion from the site of its synthesis to some other site. So everything is clear about biosynthesis of ethylene, or is there any confusion to you? If there is any confusion, then do tell me. Okay, if not, then let's move on to our next topic, which is what are the physiological effects of ethylene. So what are the physiological effects of ethylene? See, this ethylene, just like some other hormones, it can also induce flowering in plants. Gibberellin can induce flowering, auxin can induce flowering. It can also induce flowering such as mango and pineapple, in plants such as mango and pineapple. This ethylene, just like that of auxin and cytokinin, it can induce femaleness in monoecious plants having separate male and female. It means plant is monoecious but flower is not bisexual. Flower is unisexual. It will have either stamen or pistil. So plant is monoecious but flowers are unisexual. So in monoecious plants, ethylene can induce femaleness. For example, cucumber. This ethylene is also responsible for the formation of root hairs and adventitious roots. Okay, so Auxin is responsible for preventing abscission. How auxin is able to prevent uh, this abscission? Actually, auxin the act on the cells in the abscission zone. This auxin makes the cells in the abscission zone insensitive to ethylene. Okay, if the cells are insensitive to ethylene, they won't be undergoing any abscission. But when the auxin concentration is low, at the time when the auxin concentration declines, at that time these cells in the abscission zone become sensitive to ethylene and the abscission is promoted. Okay, so that's how ethylene causes the abscission. So ethylene can cause abscission when auxin concentration is low. Okay, abscission is caused by ethylene only when auxin concentration is low. If auxin concentration will be high, then cells in the abscission zone will be insensitive to the ethylene. Very important function of ethylene, and the most common one is ripening of fruits. It also causes apogeotropism. Okay, along with the ripening of fruits and many other function, ethylene also called apogeotropism, that is, it can reduce the sensitivity to gravity. We know that auxin is the main hormone responsible for apical dominance. And how auxin causes apical dominance? Auxin stimulate the strigal lectins, which is activate branched 1 genes. Branched 1 genes are responsible for the product that will result in the apical dominance. Okay, but along with auxin, ethylene is also promotes apical dominance, and it increases senescence, which is the aging effect. Okay, and we also know that gibberellin is the main hormone responsible for seed germination. But here, see the physiological effect of ethylene. This ethylene can also help in seed germination. I forgot to mention one another point, that is, it can induce epinasty. If you don't know the meaning of epinasty, do comment in the comment box. I'll tell you the meaning of epinasty as well. Okay, okay, all right, okay, fine. So these are the function of ethylene, but it can we know that internodal elongation is the function of gibberellin. But when you are asked internodal elongation in deep water rice plants, which hormone is responsible for this function? Ethylene is the one which is responsible for the elongation of leaf bases and internodes in deep water rice plants, so that leaves remains above water. If you have seen rice fields, you know that they are filled with water. Okay, especially during the initial phase of growth of plants. Okay, that time fields are completely filled with the water. So it needs that the water plants need to be remained above the water level. Okay, at that time their internodes will be elongated. And if you ask such kind of question like internodal elongation as the function of, we are immediately going to answer it's gibberellin. But remember, in case of rice plants, it's ethylene. Ethylene is a promoter as well as an inhibitor, but it is largely a inhibitor, and it is responsible for triple response. Now, what is this triple response? What do you actually see during triple response? Okay, so in triple response, we see reduced stem elongation, or we can say short hypocotyle or shoot. Okay, sometimes instead of hypocotyle, epicotyle word is used. But hypocotyle and epicotyle are both the parts of our embryonal axis. And the part of embryonal axis above the level of cotyledon is called epicotyle, and the part of embryonal axis below the level of cotyledon is called hypocotyle. But ultimately, embryonal axis is changed into a stem, because epicotyle terminal will be converted into shoot, which is our plumule. Plumule will be resulting in the shoot formation, and radicle results in the root formation. Fine. So there will be short hypocotyle, and sometimes you might have been provided with the option which includes short epicotyle or short shoot. So first effect of triple response of ethylene is a reduced stem elongation or short hypocotyle or shoot. Another function is increased stem thickening. So we can say that ethylene response may result in the thickness of stem. And next one is curled apical hook formation, or you can say apical hook formation, or horizontal growth habit, or tightening of apical growth. So you can slash here and add a apical hook formation. Okay, these are the same things. You get it or not? Okay, so these are all the physiological effects of ethylene. Do compare the ethylene physiological effects with other phytohormones so that you will be able to remember all the physiological effects of all the phytohormones. It will become easy for you to remember all these and then choose the correct option. Fine. So in the last section, we are going to talk about the signaling pathway of ethylene. Okay, the very important one. So many a times question is asked from the signaling pathway of ethylene. Okay, so remember the signaling pathway of ethylene. Don't get confused with anything. Ligand in case of signaling pathway of ethylene is definitely ethylene. Receptor is ethylene response 1. This ethylene response 1, ETR1, is actually a copper ion based receptor which is present on the endoplasmic membrane. Okay, endoplasmic membrane will have an luminal side and extraluminal side. The extraluminal side is also known as cytosolic side. Okay, so ER membrane is having the receiver domain in the lumen. Okay, so remember ETR1 is present on endoplasmic reticulum and its receiver domain occurs in the lumen of ER, and it is a copper ion based receptor. As we know, this receptor is a copper ion based receptor. There might be some transporter of copper ion. So RAN1, responsive to antagonist 1, is the transporter of copper ions. So there are also other receptors in Arabidopsis. Along with ETR1, there is ETR2, which is ethylene response 2, ERS1, ethylene response sensor 1, ERS2, ethylene response sensor 2, and EIN4, ethylene insensitive 4. So these are all other receptors in Arabidopsis. So almost 5 types of receptors are found in Arabidopsis. And next we will see the regulators of pathway. ETR1 is a receptor itself, then ERF1, ethylene response factor 1, CTR1, constitutive triple response 1, EIN2, it's what is it? Ethylene insensitive 2, EIN3, ethylene insensitive 3, and ETP. You can write it down. EIN2 targeting proteins. What is ETP? It is EIN2 targeting protein. Fine. Remember the receptors of signaling pathway of ethylene, or we can say ethylene receptors, are homologous to bacterial 2-component receptor, and binding of ethylene inactivates receptor, that is receptors are negative regulator. What does it mean? It means receptors, if ethylene is not it doesn't bind with its receptor, then it will result in the repression of ethylene responsive gene. So receptor is a negative regulator, and binding of ethylene inactivates the receptor, and after that ethylene responsive genes can be transcribed and translated further. Okay, so let's see it through a diagram. This is the endoplasmic reticulum membrane. This is our ethylene response 1, and it is having 2 domains. Histidine kinase domain, it also possesses histidine kinase domain. It will have a receiver domain. Okay, then copper ion will bind here. then after binding, there will be phosphorylation in the absence of, this thing is happening in the absence of ethylene. After the binding of copper ions, there will be phosphorylation, autophosphorylation of the CTR1, and it will result in the phosphorylation of CTR1. This CTR1 then phosphorylates EIN2 present in the ER membrane itself. Okay, CTR1 then phosphorylate EIN2. After phosphorylation, this EIN2 becomes sensitive for EIN2 targeting protein. This EIN2 targeting protein is a F-box protein which will cause ubiquitination of this EIN2, and after ubiquitination, EIN2 will be targeted to 26S proteasome and it will be degraded. Now see, EIN2 is the main protein responsible for stabilization of EIN3. EIN3 is present in the nucleus. This EIN2 is degraded now, so EIN3 will not be stabilized and it will be degraded as well in the 26S proteasome. Okay, and there will be no transcription of ethylene responsive genes because EIN3 is the one which was going to play the function of transcription of ethylene responsive genes. But just because ethylene is absent here, the copper will bind to the receptor, and then there will be autophosphorylation of receptor, then it will phosphorylate CTR1, which will phosphorylate EIN2, and EIN2 will be targeted by EIN2 targeting protein. It will be ubiquitinated and will be targeted to 26S proteasome where it will be degraded. If EIN2 is degraded, it will not be able to stabilize EIN3, which is responsible for the transcription of ethylene responsive gene, and this EIN3 will be degraded. There will be no transcription of ethylene responsive gene. Then, in the presence of ethylene, this ethylene will bind to ETR1, and there will not be any autophosphorylation. No autophosphorylation means CTR1 will be inactivated. This CTR1 is inactivated, then it no longer will be able to phosphorylate EIN2. And if EIN2 is not phosphorylated, then it is clipped off and the cleaved part or seed domain is transferred to the nucleus where it can stabilize EIN3. This EIN3 along with the ethylene response factor will be responsible for the transcription of ethylene responsive gene. This is all about the transport this is all about synthesis, not transport, but this is all about signaling pathway of ethylene. Hope everything is clear to you. I have tried my best to give you everything in a single video, which is approximately of 25 to 30 minutes. Okay, that is all for today. In next lecture, we are going to discuss the signaling pathway of our different plant hormones, which has been left like abscisic acid, gibberellin, cytokinin, and auxin. But before leaving, please let me tell you that you can, if you want to prepare for CSIR 2021, June 2021, then you can take the subscription of an academy where you will also see Neha Taneja ma'am, where she is an educator on an academy and teaches very well. Here is the detailed paid, this is the details of paid classes. You can see your fee structure here. To get the 10% off, use the code NEHA. Okay, this is Neha ma'am's code, NEHA. Fine, you can use this code. Please do not use any other code, use this one. Okay, you will also have other educators there and they all will be working for you to complete your course our syllabus for June 2021. They will help you in cracking your test, but we are also helping you in cracking your test. So do subscribe to our channel as well, and press the bell icon for getting the notification. Check out the description box where you will get the description, where you will get the links for different platforms of Gurmantra Shiksha Ka Granth. So this is all for today. We will meet you in the next lecture with new topic. Till then, take care, stay safe.